Clonal genetic diversity and its correlation to cancer risk in Barrett's esophagus. Barrett's esophagus is a condition caused by repeated exposure to stomach acid, which results in lesions. About 10% of patients experiencing chronic acid reflux will develop Barrett's esophagus, in turn increasing their risk of developing esophageal cancer. Although less than 5% of patients with Barrett's esophagus will develop esophageal cancer, it is important to determine which patients are at risk so that early detection and treatment can be possible. We hope to be able to determine whether or not a given patient has an elevated risk of eventually contracting esophageal cancer by performing a single test focusing on specific biomarkers. In order to address this goal, a group of researchers in the Netherlands, led by Pierre Martinez and Marguerite Timmer, studied how clonal expansion and diversity might contribute to cancer risk in BE patients. Samples of cells from esophageal lesions were collected from 320 Barrett's patients who had received therapy for acid reflux and who had tested negative for dysplasia. Patients were monitored for a median of 43 months, with a range of 11 to 130 months. A second endoscopy was conducted on 195 of these patients, with a median of 37 months between the two samples. From each sample, a minimum of 50 cells were to be counted and examined for genetic abnormalities. The samples were collected using endoscopic brush cytology, and then were examined using multicolor fluorescence in situ hybridization analysis. Using the FISH method, 7 predetermined genetic markers were labeled with color to identify clonal presence, size abundance, and diversity. Clonal expansion only occurs an average of once every 36.8 patient years, at a rate of 1.58 square centimeters per year, translating to an average growth rate of 0.6% of the cell population per month. Interestingly, clonal expansions were not correlated with risk of cancer. One crucial finding of this study was that the genetic diversity of a given lesion remains relatively stable over time, and that baseline diversity correlates with risk of cancer. In other words, cancer risk is predetermined by the level of diversity that exists within a patient's cells. So, the higher the diversity, the greater the risk, and vice versa. A dynamic equilibrium of clonal expansions and contractions was observed in the lesions over time. Dynamic equilibrium is maintained when there is a normal, invariant level of genetic diversity. As a result, the rate of mutations does not put the patient at high risk for developing cancer. When genetic diversity is high, however, dynamic equilibrium fails to be maintained, and mutations occur at an abnormal rate, increasing cancer risk. These findings are significant because they've indicated it may be possible to use a single genetic diversity test to accurately predict a BE patient's risk of developing cancer. Because the risk of cancer remains constant for at least 3 to 4 years, the test would not need to be repeated often. Diversity tests using specific markers would allow doctors to monitor at-risk patients so that progression to cancer could be detected early, and less invasive treatments could be used, potentially increasing the patient's chance of survival. Dynamic equilibrium and genetic diversity are the foundation of the naturally occurring process of evolution, which has shaped life on Earth ever since the appearance of the first organisms. We have learned that in order for evolution to occur, genetic variation must be present. This variation is caused by mutations, which can disrupt the previously existing allele frequencies. Higher rates of mutation increase the likelihood that a cell can become cancerous, explaining why increased genetic diversity is correlated with higher risk of cancer in BE patients. If a cell develops an advantageous mutation, it will survive to reproduce, and in the case of cancer cells, this is often at the expense of the surrounding cells. In this experiment, mutations were found in almost every patient, often at the P16 locus. The role of this and other specific gene locations in esophageal cancer shows how a single mutation can disrupt the entire structure of a cell, which can in turn disrupt the functioning of the body as a whole. We may never fully understand the complexities and implications of human genetics, but experiments like this one are constantly bringing us one step closer.